So first of all, declarations of interest. I do have some, and what I'll do is flag those up when we get to the relevant point in the talk. What I thought I'd do in the next half an hour is give you sort of an overview of circadian rhythms and sort of the biology of sleep and circadian rhythms and its relevance and why this dynamic physiology is potentially so important. Then talk a little bit about the regulation, the entrainment of the circadian and sleep-wake systems by light, and flag up some, I hope, some interesting information, but also deal with at least one misconception. Then talk about the impact of sleep disruption generally across health. And if there's time, then finish with a project that we're involved in called the Teen Sleep Project, which is the development of an educational intervention to try and help sleep in young people, and the potential for that, I think, throughout school systems, indeed around the world. So if we kick off with circadian rhythms and overview, and to state the breathtakingly obvious, we sit on a planet that revolves once approximately every 24 hours. And this generates a very complicated ecological environment, not least because of the profound changes in light, in temperature, availability of food, and all the rest of it. And as a consequence of these profound changes, we, and indeed most life on the planet, has evolved a behavioral response, which in us is defined as the states of sleep and consciousness, two profoundly different behavioral states. And these different behavioral states are underpinned, no great surprise, by very different sorts of physiology. Now, what you see in each of these blocks here is a 24-hour time base, and this sort of gray area here is approximately when one would be asleep. And let's first of all look at the changes associated with core body temperature. And we see that during the day, core body temperature is relatively high. In anticipation of sleep, it drops. And then in anticipation of wake, it rises. And the key point about this parameter and all the other parameters I'm going to be talking about is that these persist under constant conditions. This dynamic change is endogenously driven by a circadian clock, a circadian rhythm. We've seen an example here of core body temperature. We can look at a whole range of other parameters. So, for example, we'll talk a little bit about systolic blood pressure later on. Alertness is very interesting. A big drop in alertness, our ability to function at 4 o'clock in the morning here, is as impaired as if we'd consumed sufficient alcohol to make us legally drunk. So your ability to process information at that time of day is markedly impaired. It rises, of course, in anticipation of the need to be alert and awake in our environment. Growth hormone, as many of you will be aware in this audience, has this sharp rise during the first half of the sleep phase. But what's interesting about growth hormone is that it's also modulated by sleep processes itself. So if you don't sleep, then this spike, this early early rise in growth hormone is significantly attenuated in the absence of sleep. We see, of course, cortisol rising during the early hours of wake and then declining a little bit. And then, of course, the biggie, the most obvious of our 24-hour behaviors, sleep, which is what we'll talk about in a little bit more detail. But first of all, let's consider the generation of these circadian rhythms. And as many of you will be aware, I'm just going to use the pointer again. You can't hear. Is that better? Yeah, sorry. So as many of you will be aware, in the anterior hypothalamus here, there's an extraordinary structure called the suprachiasmatic nuclei. It's often called the master biological clock. And the SCN, as it's mercifully abbreviated, comprises about 50,000 individual cellular oscillators. So if I were to take one of those cells out and put it in a dish, you would still see a 24-hour oscillation in electrical activity or indeed gene expression. And what's become clear over the past few years is that this cellular clock is based upon what's called a TTFL, or transcriptional translational feedback loop. Essentially, a gene produces a protein. The protein then negatively feeds back on the gene's production. There are about 20 to 30 of these key clock genes. But the net effect is to produce a 24-hour oscillation in clock proteins, and this provides the essential signal that drives the rhythmic behavior that we were talking about earlier. Well, it's no good having a clock unless it's set to the external world. Oh, yes, and I should have said, sorry, these 50,000 individual cells here, these individual cells are coupled together to form this sort of single circadian oscillator. And that's of absolutely no use at all unless the internal clock is set to the external world and its exposure to the light-dark cycle via the eye that provides this critical entraining cue. If it weren't for that, then we would, for example, drift through time. Classic mismatch between the internal clock and the external world is jet lag. We eventually get over jet lag, as some of you may be experiencing, as a result of exposure to the local light-dark cycle. Okay, well, what we thought used to happen was that the eye, setting the clock, would then provide messages which would drive rhythmic physiology in the rest of the body, and those sorts of physiological changes that we saw. We got that wrong, and what's now very clear is that every cell in the body has the capacity to generate a circadian rhythm. So what seems to be happening is that it's not the clock forcing rhythmicity upon the rest of the body, it's coordinating rhythmic activity. But it's got a bit more complicated. So, for example, these peripheral clocks can respond to other signals other than the master clock and the SCN. So, for example, food seems to be an important entraining agent to the liver and the gut. Via probably signals involving glucocorticoids. And although I've emphasized the role of the light-dark cycle, I should also say that the sleep-wake cycle itself, so for example, the longer we're awake, the higher the levels of adenosine that rise within the body, within the brain, and that adenosine signal can also modulate the effects of light on the clock. So there's an interaction between these various agents, which essentially then allows us to align physiology and behavior appropriately to the external world. As I say, dominated by the light-dark cycle, but also local modulators. And so we see here, a beautiful circadian-driven rise in melatonin, cortisol, and again in growth hormone. And the whole point of this is that the circadian system can fine-tune physiology and behavior to the varied demands of the night-day and the sleep-wake cycle. Okay, so what are some of the consequences of this dynamic physiology? And I raise some general points here that I think we should all consider, whether we're experimental scientists or clinicians. And the first is something relatively straightforward. This is systolic blood pressure, and we've seen this figure before, and we showed that systolic blood pressure is high during the day and drops to low levels during the night. And this is an individual who has healthy systolic blood pressure, but some individuals will have high systolic and, of course, diastolic blood pressure. So even in those early hours of the morning, you can have 100 milligrams of mercury blood pressure for systolic blood pressure. And it's this sharp rise with high blood pressure that seems to be really important. And these are some data from my colleague Peter Rothwell. And what he's plotted here is time of day on this axis and the frequency of a stroke on this axis. And what he's shown is that between 6 a.m. and 12 noon, there's a 50% greater chance of having a stroke than any other time of the day. And this, of course, is related to this very sharp rise in endogenously driven blood pressure. Now, these sorts of observations, I think, raise two important points. First is when to take our stroke medication. So if for many of us, we were taking our stroke medication during this peak window here, whereas ideally we should be taking our stroke medication here. And we have yet really to start to deliver drugs at the appropriate time of day to intersect with when they're actually needed. The other, and I should point out, this is a reminder for me to mention to you, is that most drug and cognitive testing, of course, is undertaken on nocturnal rodents. So what we're doing is waking up a nocturnal animal in the middle of its sleep phase and doing some observations, and then extrapolating that to our wake phase. And of course, that can be a profound difference. So toxicity, for example, can vary from 20% to 80%, depending upon time of day in rodents. And there is an argument that many of the drugs that we are currently that we have abandoned should actually be retested to see if they would be more effective at a different time of day. The other implication other than when to take our medication, is how to prepare our healthcare services, knowing that there's going to be that high window of stroke at this particular time of day. Are our ambulance crews and our hospitals adequately prepared to deal with that rush of stroke? Another area, which is an expansion of the points I've already made, which is the timing of drug delivery, or this whole field of chronotherapeutics. And I want to just give you one sobering and one interesting example. This is a recent paper, 2016, which looked at the impact of whole brain radiotherapy on survival in patients that were exposed to the radiotherapy at 3 different times of day. In the morning, 8 a.m. to 11 a.m., 11 a.m. to 2 p.m., or 2 p.m. to 5 p.m. And these are the results. The morning group were all dead by about 6 months. The lunchtime midday group were all dead by 24 months, but a significant difference between the two. And the afternoon group, 10% were still alive by 36 months. So same sort of treatment, different time of day, has had a big impact upon long-term survival. And this is something that when anybody takes the time to look at time of day effects on drugs and interventions, you always see big differences. This is a particularly dramatic example. Another important example, I think, is within the context of vaccination. This is an example we saw where morning treatment was a bad time for radiotherapy. These individuals who had their influenza vaccine in the morning had a titer that was 400-fold, or a mean antibody titer of 400, versus those that had in the afternoon with a titer of 100. So again, time of day has had a big effect upon the antibody titer, the result of vaccination. To my knowledge, but maybe somebody here can correct me, I don't know of anybody who's done this in young people and the impact of time of day on titer for some of the key immunizations that take place in our young children. Okay, so the point I wanted to make during these early slides was that time of day profoundly matters. Let's now move to the most obvious of our 24-hour rhythms, and that's the sleep-wake rhythm. And let's consider the biology of sleep and circadian rhythms. Now, what's fascinating, and I always find this quite extraordinary, is that if you think about across the lifespan, 32 to 36% of our entire lives will be spent asleep. And we, as a society, marginalize it and dismiss it. We've been kind of studying sleep for 60 years, but that's been primarily through EEG. And I don't mean to be dismissive of this work, well, I guess I do, but not very publicly, is that we've been recording these sorts of wiggles for 60 years. And of course, we can define different sleep states, and we can define a REM and a non-REM sleep. But it hasn't told us anything fundamental about what's going on within the brain whilst we sleep. So we have to sweep ahead to the last 10 to 15 years to get some understanding of what critical processes are going on within the sleeping brain, and indeed why sleep is so incredibly important to our biology. I want to first concentrate on brain function and consider the consolidation of memories whilst we sleep, the fact that the tiredness state of the brain will define what sorts of things we remember, and indeed the processing of information, not just the retention of facts, but actually manipulating that information whilst we sleep. Okay, so let's now consider information processing, and this is a beautiful study, quite a famous study from Jan Born's group, published way back in 2004. And what Jan's team did was develop a problem-solving task. And in this particular group here, he introduced them to the task in the morning, and then they performed the task in the afternoon. And you see here that 20% solved the problem. They sorted it out. The next group, he introduced the task in the morning, but then they performed it the following afternoon, but they were deprived of sleep. And what we see here is that about 20% solved the problem. So he'd nicely controlled for fatigue. Of course, this is the interesting group. Introduced the task to them in the morning. They performed it the next afternoon, but with a normal night of sleep. And 60% or more actually solved this problem task. And this illustrates, I think, absolutely beautifully that sleep promotes the ability to come up with novel solutions to complex problems. And boy, do we need those. The other area I'd like to mention is sleep, memory, and emotion. And this is a beautiful study from Sickold's group, and again published, I think, in 2006. And what we have are two groups here. There's a group who've slept normally, and then another group who were deprived of sleep for one night of sleep, but they hadn't slept for 36 hours. And these two groups were asked to remember words with a different emotional content. So positive words, joy, happiness, love, all that kind of stuff. Negative words, hate, war, crime, Brexit. Sorry, cheap shot. Or neutral words, words something like cotton. So let's look at the words all lumped together. So we see here that this is all relative to each other, normalized. So the normal sleep group versus the sleep-deprived group, and we see a significant failure to remember all of the words, so emphasizing the fact that sleep helps the retention of the establishment of memory. Now let's break the words up in terms of their emotional content. So the neutral words, there was a trend to remember fewer neutral words, but it wasn't significant. There was a trend to remember fewer negative words, but it wasn't significant. The most extraordinary result, I think, was this huge failure to remember positive words. So tired people remember the bad stuff, but forget the happy, good stuff. And again, we might be able to reinforce that with anecdote. Okay, well, in addition to these important brain functions, the brain is also regulating the removal of waste products, as we will be, you're aware from this meeting, the regulation of growth and repair and tissue repair, the replacement of energy reserves, and rebuilding of metabolic pathways. Essentially, our ability to function during the day is utterly dependent upon the biology that's going on at night. I just want to draw your attention to one feature here, the removal of waste products. This is some stuff I was, I suppose, a bit skeptical of when it first emerged, and that was sleep helps you get rid of beta amyloid, this misfolded protein associated with dementia and Alzheimer's. And it was early work, but the most recent data, I think, is pretty convincing. There was a study published fairly recently showing that one night of no sleep would increase levels of beta amyloid within the CSF by 3%, which is, I think, an important finding. One certainly wouldn't say that no sleep is going to cause dementia or Alzheimer's, but it might be a significant contributing factor. Okay. Well, in view of the amount of time spent asleep, across the lifespan, as I said, 32 to 36% of our entire lives, and in view of the fact of all the important things going on within the brain, it's no great surprise that sleep arises from an interaction of all the key brain neurotransmitter systems, and this is important, and we'll come back to this later when we talk about mental health, and also an interaction of multiple brain structures. So we have the hindbrain, we have the hypothalamus, the midbrain, and the cortex all interacting to produce these extraordinary states of consciousness and sleep. So the point I'm making is that sleep is a global brain event. That's fine, but what we've also got to consider is the timing. How do we get this switch from sleep to wake? And that involves the clock, and we've already touched on that, another process called sleep pressure, or the homeostatic drive, and then, of course, societal pressures of work and family. Let's look at these interactions on regulating the sleep-wake cycle. Okay, so here we have what we've seen before, the eye regulating the circadian system, and what the circadian system is doing is essentially time-stamping our physiology. Now is the appropriate time to be awake, and now is the appropriate time to be asleep. And that critical function interacts with the sleep pressure or the homeostatic drive for sleep. And perhaps the most intuitive part of sleep is the longer you've been awake, the greater the sleep pressure, the greater the need for sleep. And let's now see how sleep pressure and the circadian timing systems interact. So here we have a wake-sleep cycle, and I'm just going to first of all talk about the sleep pressure, and so we see sleepiness along this axis here. And this sleep pressure has been associated with the buildup of adenosine within the brain. So what happens is that longer we're awake, there's a rise in sleepiness, sleep pressure, And then when we sleep, this sleep pressure is dissipated. But late afternoon, early evening, our sleep pressure is really high. But you don't fall asleep because of the action of the circadian system. Essentially, what the circadian system is doing is providing a drive for wakefulness. So as we go through the wake state, the circadian system sort of keeps up with that rising sleep pressure by saying, stay awake, stay awake. And so the circadian drive for wakefulness is really high towards the end of the day, in the evening, because it's compensating for this high level of sleep pressure. As we approach sleep, the wakefulness drive diminishes, and then we go to sleep. The sleep pressure exceeds the wakefulness drive from the circadian system. And then the wake pressure is low throughout much of the sleep episode, and we experience a series of REM, non-REM cycles of sleep, and then ideally we wake from REM sleep, again when the wake drive exceeds the sleep pressure. So these two systems interact beautifully under normal circumstances. They're completely screwed up in night shift workers, for example, and in other individuals where societal pressures force us to work against our internal biology. And it's also a pressure that as we'll see later, adolescents also face. One other thing I'd like to mention is the use of caffeinated drinks. Why caffeine is so effective at keeping us awake is because it blocks the receptors that respond to adenosine and the sleep pressure. So you take a cup of coffee, you block those adenosine receptors, and the brain thinks it's less tired than it actually is. So that's how caffeine is working in this system. Okay, let's now move on to the role of light and the eye. So this is entrainment by light. And as we discussed, the eye is providing light information to drive entrainment of the clock. And let me just be clear what I mean by this. So here's midnight to midnight. This is darkness and this is light here. And what I mean by entrainment is that essentially the sleep will occur on subsequent days at about the same time. We sleep about the same time because the circadian system is entraining the sleep-wake cycle. Now, if we resided in complete darkness, so went down to a deep dark cave, then the clock would still tick, but in humans, the clock is slightly longer than 24 hours, and so it'll drift with time, so we'll get up later and later and later. And that's called free running. And of course, this is a big problem, as we'll touch on later, to those individuals who have no eyes. They have no way of setting the internal clock to the external world, and they spend their lives free running. Okay, so it's the eye, as I said, but what's turned out to be truly extraordinary, and it's work from our lab for now 20 years or so, addressing the question, how does the eye regulate this clock here? And let's strip back the eye to expose the retina. Here we see the rods and cones, the inner retina, and the ganglion cells that give rise to the optic nerve. And what turned out to be extraordinary is that you don't need your rods and cones at all to detect that light. So in rodent models, and subsequently in humans and rats and primates, it's been shown that the rods and cones are not the photoreceptor detecting the light-dark cycle, but there's a third receptor system within the eye based upon a relatively small number of ganglion cells. About 1 to 2% of the ganglion cells are directly light sensitive. These are photosensitive retinal ganglion cells, and they are sensitive, maximally sensitive to blue light, and this is based upon a novel photopigment which has been called melanopsin, or OPN4. And this is the basis for the sensitivity, the blue sensitivity that we'll talk about later, and many of you will be aware of. So the point I'm trying to make is the eye provides us with both our sense of space, through the visual system, of course, but also, as a result of the regulation of the clock, our sense of time. Here we see an example of an individual, and this is a 48-hour time base. A 48-hour time base. Here's midnight, and this is a student, and so they're going to bed a bit late, and they have a fairly ragged get-up time. But you see that it's broadly entrained, and this contrasts with an individual who has lost their eyes later in life, and you see that their rest activity, their activity is drifting through time. And that's what happens if you don't have eyes. And we're using our knowledge of how light and sleep regulate the molecular clockwork to correct circadian rhythm disruption, ultimately to try and help those people with profound visual damage, eye loss, or profound damage to the eye. And here is where I said I would declare my potential conflict. I am a co-founder of this company, Circadian Therapeutics, and some of the work I'm going to talk about now has been funded by venture capital that has been invested in this company. Okay, so here's our depiction of the retina and those photosensitive ganglion cells projecting to one of those SCN neurons. And what we've been doing has been to work out essentially what's going on. What are the signaling pathways whereby light detected by those receptors, here we go, is actually shifting the clock. And where we are at the moment is that we know what those signaling pathways are, and so we've been able to develop drugs which can also shift the clock based upon the same pathway and essentially fool the clock that it's seen light. Okay. So what we've got here is a pharmacological replacement of light experiment. And just to remind you what we're doing, we're going to look at light and its shifting effect upon the clock. And then we're going to look at the impact of two drugs and the shifting effect upon the clock. Right, here's the control, and it's been normalized to no change, and we see here a change in the clock on this axis here. The first thing is we're going to look at the effects of light, and we see, no great surprise, light will shift the clock beautifully. The next drug, now we're going to use a drug called talsimeltion. Talsimeltion is the only FDA-approved drug to be administered to individuals with non-24-hour sleep-wake disorders. It's not very effective on mice, and indeed in humans it's not very effective either. But there's nothing else on the market at the moment. This is one of the drugs that we have been working on. It's an adenosine antagonist. And we get a beautiful shift of the clock, which is not significantly different from light, and therefore highly significantly different from our control. So we are very optimistic from these mouse studies that we can then move on and start to work with humans. And the first patient group we're working with are the Blind Veterans UK, individuals who've lost their eyes as a result of trauma and showing that drifting circadian rhythm. Can we give them back a sense of time? And because this is a repurposed drug, we can move very quickly from the mouse to the patient within months. Okay. So that's one of the avenues that we're exploring. Another important point to make is the timing of light exposure. So here's again a light-dark cycle, and if you see light at dusk, as illustrated here, you will delay the clock. If you see light at dawn, as illustrated here, you will advance the clock. Now, when we were agricultural workers, we got a symmetrical pattern of light exposure at dawn and dusk, so we basically were on track. And I just want to show you a study that we published last year, I think it was, led by Kate Pochette, which was looking at morningness versus eveningness in a student population. And what we discovered, in short, was as follows. So the students were sleeping through dawn, so they were not getting dawn light exposure. But of course, they were waking up late and then getting dusk exposure. So the net effect was exposure to light at dusk, which delayed the clock, without a commensurate exposure to light in the morning, which would advance the clock. So part of the problem that young people face in the student body is that they're getting a dusk light exposure without dawn light exposure, and that's pushing them to the clock to a later phase. So it's really important to get light exposure at the correct time for appropriate regulation of sleep-wake timing. How much light for entrainment? And this is also an area of controversy. Here we see the broad light exposure in the environment from very dim light to full sunlight. And of course, the rods are providing us with our dim light exposure, the cones giving us our color vision in this range here. But those photosensitive retinal ganglion cells are remarkably insensitive to light. They need hundreds to thousands of lux to get a robust light signal. So most of the time, we're exposed to light levels, 300 to 400 lux, that would be not sufficient to robustly entrain the sleep-wake cycle, the clock, to the external world. And so seeking out natural light and bright light is really important for that process. And this raises the point about what about evening light exposure for computer screens? There's endless discussion about this in the press, and that the light from computer screens is shifting the biological clock. It's producing about 40 lux, which isn't sufficient light to shift the clock. And so we've got these devices which turn blue screens into red screens, and are reported to not stimulate those photosensitive retinal ganglion cells. They weren't being stimulated anyway. And kids come and tell me, say, it's fine, I can use the screen up until whenever, because I'm not going to shift my biological clock because I've got this device that turns the screen red. Yes, that may be true, but they're still up at 2 o'clock and 3 o'clock in the morning using the devices. And so it's not the phase shifting effect of light from those screens, it's actually the duration. It's not the light, it's the duration of use that's leading to shortened sleep and tiredness in teenagers. Okay, and one last bit of data on light, and we're close. Okay, my clock hasn't changed. You see, it's still saying 25 minutes here. So that means, how long have I actually got then?
1 minute.
1 minute, okie dokie. Right, then I'll give you this really exciting bit of data about light in non-seasonal depression. It's not our work, but I do want to draw your attention to it. Weeks along this axis, and then here's the change in depression, a reduction in depression along this axis. And the data looked at a placebo, and you see there is a placebo effect in reducing levels of depression. What's fascinating is that fluoxetine, Prozac, was significantly effective after only 8 weeks. Light, however, and light was 30 minutes in the morning at 10,000 lux, was significantly effective at 2 weeks and was robustly effective throughout the rest of the experiment. But what was fascinating is that light and fluoxetine together was additive, suggesting that light and fluoxetine are operating on different paths. Okay, so I've massively run out of time, and I need to stop at this point, I guess. Sorry about that. What I wanted to sort of allude to, I'll just show you one last slide, is that sleep disruption is so much more important than the inconvenience of feeling tired at an inappropriate time. We see loss of attention and high levels of microsleeps. We see feelings of stress and anxiety, difficulty to wake up or fall asleep, impulsivity, that negative focus we talked about, impaired memory that we talked about, increased mistakes, reduced cognition, creativity, and information processing. And these are the sorts of short-term effects that 1 or 2 nights of sleep loss can precipitate. Sustained sleep loss, as in adolescence, the business community, and various other sectors, is associated with increased stimulant and sedative use, stimulants driving the wake of day, sedatives to reverse that at night. There's immune suppression, probably because of elevated levels of cortisol. That elevated cortisol and reduced immunity may be the basis for increased infection and cancer risk in very long-term night shift workers, increased cardiovascular disease, risk of diabetes too, massive changes in the metabolic axis and indeed indigestion. And then finally, sleep, both short-term and long-term sleep disruption, can have a big impact upon mental illness vulnerability. So, for example, sleep disruption in those who are vulnerable give rise to mood instability, increased levels of anxiety, paranoia, and hallucinations, and then an exacerbation of the symptoms of bipolar and schizophrenia. So we've got to take this seriously. And what I would have finished with is some exciting new data by using an educational intervention in young people to give them the educational tools to improve their own sleep. And what we've shown is that educational interventions alone can significantly improve the levels of sleep in adolescents. And the next phase, of course, will be to use those approaches and then do a long-term study to see if it improves conditions like metabolic abnormalities, obesity, long-term educational outcomes, and indeed as a long-term biomarker, if you like, of mental health problems. And with that, I'll finish. Thank you for your attention.